So I think that we can get started. So for those of you who are online, you may know that we're doing a series of webinars to try to share information on the different indicators that are in the Global Biodiversity Framework, monitoring framework. All of the information on the methodologies for these indicators are available online on the SAPSTA 26 website. So I think we can put that in the chat, and you can then find the different metadata for the different indicators. There are 27 different headline indicators that have methodologies that have been recently developed by the ad hoc technical expert group on indicators. These have been developed with a wide range of partners, including for this particular indicator with GeoBon and with the GBike Consortium. They've been working for many years on indicators related to genetic diversity. And so the effort of the ATAG has been to make sure that this is presented in a way that's useful for countries in order to complete their national reports and to report against the CUNMI-Montreal Global Biodiversity Framework. As I mentioned, we're doing a series of webinars. I'll also put the link in the chat so that those of you who are interested can join us. We'll have one tomorrow on restoration and then another one on the global ecosystem typology this week. And I'll let you check the schedule after that. So with that, I'd like to introduce the fantastic panel that we have, or at least a few of the people. So Katie Millett is the Executive Secretary of GeoBon, the Group on Earth Observations Biodiversity Observation Network. They are a group of researchers dedicated to improving the delivery of biodiversity information. The GeoBon is represented in the ATAG. In fact, the co-chair of GeoBon is also one of the co-chairs of the ATAG. And the other co-chair of GeoBon is also a member of this ATAG on indicators. And there's a number of indicators that really build from essential biodiversity processes and work that's been done by GeoBon. And then Belma, who is also with us, is a member of the ATAG. She is representing Bosnia and Herzegovina, and she's taken the chaperoning of this process for this particular indicator on, and she'll be the moderator for this session. So I'll hand over to you, Katie.
Thanks, Jillian. So welcome, everyone. It's my pleasure to welcome you to this webinar on reporting and monitoring genetic diversity with indicators that do not require DNA data. So as Jillian mentioned, we have 3 speakers today, and these presenters have been working on these indicators for 4 years. They've been receiving input from parties and stakeholders and working continuously with them over that process. So today's webinar will provide you up-to-date information about how countries can meet the GBF's commitments to conserve genetic diversity on all species, wild and domesticated. First, we'll explain the indicators for reporting on genetic diversity, focusing on indicator A.4, which is for goal A, including what the indicator means and where data can be found. We will present the results from a pilot testing of the genetic indicators that was conducted in 9 countries. And this study on these results will demonstrate how feasible these genetic diversity indicators are using a variety of data in a rapid and affordable way. Then our speakers will explain recent and ongoing work in 2024 to support the parties. So there's a lot of new things coming to be available for this indicator, with an aim to make reporting straightforward, affordable, and useful for directing conservation action, and to receive input, suggestions, and comments from parties in order to improve the indicator and meet their needs. So we'll briefly provide suggestions for genetic diversity in the context of the NBSAPs, or the National Biodiversity Strategies and Action Plans as well. And of course, we'll have a question period where we're open to your questions and take any suggestions that you might have. So our first speaker that I'll hand it over to is Belma Kalamudrik-Stroy, and she'll give us a bit of a background on the indicator and the basics of the indicator itself and where the data comes from. So Belma, please go ahead.
Thank you, Jillian and Katie, for the introduction. I would like to, of course, greet and welcome all participants, and thank you for coming to this webinar today. So just let me share this screen. And just please let me know if you can see it.
Yep. Yes, all good.
Excellent, thank you. So as Gillian said, and Katie as well, my name is Belma Kalamoid-Strel, and I was a part, or still am a part of the AHTEG on the indicators for the CBD, and I coordinated the group within the AHTEG that was working on the indicators that we will present to you today. So, okay. So although genetic diversity and its importance have been recognized by the Convention of Biological Diversity since 1992, the implementation of the convention to safeguard the genetic diversity was lagging behind. Luckily, parties to the CBD at COP15, held in Montreal in 2022, committed to more ambitious actions towards protecting genetic diversity below species level. A historical milestone was adoption of a stronger and clearer wording within Goal A and Target 4 to maintain genetic diversity, not only for domesticated, but also for native wild species, and not only between populations, but also within populations, and all within with the final goal of safeguarding the species' adaptive potential. So I would like also to stress that the Target 4 wording also contains term restore, meaning that actions should be taken if poor genetic condition is noted within species and populations. So genetic diversity is based on DNA variation, and it represents the amount of variation within species and their populations or breeds, which can be observed in trait variations. For example, thermal tolerance, color, size, phenology, and so on and so forth. The genetic diversity helps species adapt during environmental changes, such as heat waves, pollution, emergence of new pests, diseases, or invasive species. It prevents inbreeding depression, and it makes ecosystem resilient to extreme events. It supports agriculture, forestry, and fisheries, and hence it is vital for both nature and society. So, however, genetic diversity, although important, is declining across the globe and across taxonomic spectrum. So to monitor the status of genetic diversity and track changes in diversity over time, indicators were designed that can use DNA-based data, but that can also be calculated based on proxies, so without DNA-based data. And because of this flexibility, they can actually be applied in all countries in the world. This approach allows the use of many kinds of data, making the indicators inclusive, meaning various actors can actually participate in the process, and affordable, meaning that they are useful in most of the countries, or all countries, of course, in the world. So two of the designed indicators are part of the Global Biodiversity Monitoring Framework. One that you can see on the right has been adopted as the headline indicator for the Target 4, and it assesses the proportion of populations within species with an effective population size above 500 individuals. This indicator regards within population component of the target, and parties are obligated to report on it. The other indicator, what you can see on left, is currently a complementary indicator that assesses the proportion of populations maintained within species, and it regards the between populations component of the Target 4. It looks at the number of historical population that existed versus the number of populations that exist today. It is important to stress that both indicators can be applied for all types of species. So with this presentation, we would like to show you that genetic diversity indicators are feasible at scale and applicable for all types of species. They leverage in countries' ongoing diversity efforts. They can make genetic monitoring affordable, inclusive, accessible, and useful. And they can underscore a critical conservation message. So question is, what is the effective population size, or NE for short? Simply put, it is a number of individuals in a population that mate and produce offspring, and it shows how a population behaves genetically. On the other hand, the census size represents all mature adult individuals in the population. So you see that effective size is related to the census size, but it is smaller since not all mature individuals in the populations reproduce. And in the absence of DNA-based data, which might be the case for many countries in the world due to the scarce genetic monitoring programs, the effective size still can be calculated based on the ratio with the census size. Although it can depend, of course, on the taxonomic group, the rule of thumb says that NE usually is around 10% of the census size. So if we can count the numbers of individuals, adult individuals, we can actually estimate the effective size. So the NE headline indicator actually focuses on a well-established threshold to prevent genetic erosion and maintain adaptive capacity. As you can see on this graph, much below this threshold of 500 individuals within the effective size of a population, genetic erosion is rapid. It declines. However, much above this threshold, genetic erosion is slow or minimum, and actually it's closing to zero loss. The CBD has used the concept of effective size for several decades because a threshold of effective size is actually a basis how a domestic breed is declared threatened. So distinct populations are the wild equivalent of breeds, so we are just adapting the use of this very well accepted concept. So the concept is also used in different sectors, such as forestry and fisheries. So many practitioners are familiar with it and can easily understand this concept. It's very important to stress that in several countries around the world, NE indicator has already been adopted within the national policies, and it's been used to designate whether populations are in favorable or recovering state for some amphibians, carnivores, and trees. The other indicator, proportion of distinct populations maintained, focuses on the genetic diversity between populations. As you can see on the left of this graph, of this slide, sorry, depending on the specific habitat conditions across the species distribution range, populations actually might be locally adapted to heat, cold, precipitation, and so on and so forth. And these adaptations actually reflect the genetic diversity that these populations harbor. It is easy then to understand that loss of genetically distinct populations over time can consequently mean the loss of unique and vital traits that ensure species and ecosystems resilience and functionality. So this is, of course, true for both wild and domesticated species. So it is advised that this indicator is reported along with the NE indicator. So since populations have to be delineated anyway for the computation of NE indicator, reporting on this particular indicator, populations maintained indicator, would actually not pose an additional workload for the countries. Similarly to the NE indicator, this indicator as well has been already a part of the policies and species monitoring and management in many countries. It's been used for designation and maintenance of distinct populations. In many instances, populations actually can be easily delineated even by non-genetics experts. So the populations maintained indicator actually fits very neatly into the existing policies, plans, and legislation. So currently, we have to say that there is no ready-to-use global database from which all parties can mine data to calculate these indicators, although some initiatives are already ongoing. However, we have to stress that this is not a limitation, but a strength. It actually allows countries to custom tailor the data collection processes, empowering them to address their national goals and interests. This actually approach ensures that data come from a variety of sources, fostering wide participation and inclusivity in biodiversity reporting. So leveraging on indicators' flexibility regarding the data type, data actually may come from, as I said, various sources, for example, species management reports or recovery plans, data within IUCN and red lists and national data sets, different scientific papers, or even collaborators with geneticists or the experts, local experts in the countries. The data can actually also be gathered through work with the NGOs, local knowledge holders. They can come from already existing citizen science initiatives and so on and so forth. Of course, depending on the needs and the goals of the country. And with this, I would like to thank you for your attention and conclude my presentation. And now I give floor to Dr. Jessica Da Silva, who will tell us about the testing of the indicator and the initial results. So thank you.
You're muted, Jessica.
You're muted.
Thank you. Thanks, Velma. Hi, everyone. Apologies for that. My portion of the presentation will focus on answering the questions that are seen here on the screen. When these indicators were proposed, many people questioned if there was enough information to report on species and for how many species it would be needed. And they wondered how difficult it would be to compile all this information and assumed it would come at a great cost of time and money. So to help address these questions, a group of us started a pilot project to test the feasibility of reporting on the genetic indicators. As has already been mentioned, 9 countries participated, and these countries spanned 6 continents and varied in economic status and biodiversity richness. And indeed, 5 of them were megadiverse countries. And there were over 40 people who were actively contributing to the bulk of the species assessments across various institutions, including academia, government, non-government institutions, community groups, and more. Some of them are actually in the webinar at the moment. And several others also provided information in varying capacities. And this was not just senior personnel or experts in their respective fields. Interns, students, and early career staff also participated significantly. So it was a large collaborative process that involved constant refinement with the tools and methods co-created amongst all the different country groups. And just to give you an idea of how teams worked, each country was led by about 1 to 2 people who basically oversaw, trained, and coordinated the overall country assessments. Teams were generally made up of about up to 10 people, but as I mentioned, additional people contributed as well. And it generally took teams 1 week to become familiar with all of the materials and documents needed, which Alicia will present in just a moment. And on average, it took people about 2 to 3 hours to complete a species assessment. And just to be clear, an assessment did not only involve finding and extracting information on the specific numbers of subpopulations, although that was definitely one component, but other biological information that could be used for comparative purposes and further investigations, such as distribution, threat status, and life history traits, were also compiled. And so there is room for even the 2 to 3 hours to be refined and condensed should these endeavors coincide with other species assessments. And people were basically asked to use any and all available data from whatever sources they had access to, as Velma just alluded to. Another country teams were also responsible for selecting the species to assess, and we encouraged at least 50 species, but ideally up to 100 species and more if possible. In order to truly assess the feasibility of the indicators, we asked teams to try to maximize on representativeness in terms of taxonomic groups, such as mammals, birds, fish, angiosperms, or any type of taxonomic group, ecosystem type that the species were found in, threat status, extent of distribution in terms of widespread or restricted species. We also encourage teams to select species possessing differing history traits, as I just mentioned, and ones that may be of high national importance for whatever reason, and even others that might not be. So we wanted a good spread of species. And once species were selected, teams could go out and gather their data, and once gathered, the indicators could be calculated. So here's just a simple theoretical example of how this was done. In this illustration, a fish species is made up of 4 distinct populations, and these could be associated with tributaries, for example. And one of the 4 populations, the one to the bottom right, has gone extinct. So from this information, we can calculate the populations maintained indicator. So 3 of 4 populations remain, which gives it a score or value of 0.75, or 75% of the populations remaining. Of the populations that remained, of the 3, the effective population sizes are known for each. Only one of the 3, the top right population, has an effective population size greater than 500. So from this, we can calculate the NE 500 indicator. And so that's one of 3 populations that gives you a score of 0.33 that have an effective population size greater than 500. And so country teams can calculate this for many more species across various taxonomic groups. And then at the end, the indicator values can be averaged to get a country level indicator for metric for each of the indicators. This, depending on the number of species assessed, this can also be disaggregated by taxonomic group or threat status or habitat type, whatever is most meaningful for the country. And to better illustrate this in real life, I'll just show you 2 actual examples in the next couple slides. This first one is from Mexico, and based on occurrence records found in herbaria and citizen science databases, 17 populations of this Mexican mountain juniper were identified. However, 2 of the populations were known to be extinct in the wild, so only 15 of the populations actually remain. And so based on this information, 15 of the 17 gives you a value of 0.88. So 88% of the populations are maintained. Of the 15 remaining populations, only 11 had census data that was acquired by consulting with local park rangers and botanists. And of these 11, 4 populations had over 5,000 individuals. And if we apply that default conversion ratio of 10% or 0.1 that Belma just spoke about, and it was found to be quite robust in various other studies, we can calculate the effective population size. So that 500 individuals translates to an effective population size of 500, and only 4 of the 11 were above this threshold. Therefore, the indicator value for the species is 0.36, or 36%. Our next example is from South Africa, and it's a very tiny amphibian known as Rose's mountain toadlet. The species has a very restricted distribution, and reviewing the literature, it was uncovered that 6 populations originally existed, but currently only 2 populations remain in very genetically distinct, isolated localities. And so from this, we can calculate the populations maintained indicator. 2 of 6 is 0.33, or 33% maintained. Extensive ecological and molecular studies were previously done on the species, and both were able to provide estimates of population size. So capture marker catcher data, which we are equating to census size, indicated that both populations had less than 2,000 individuals in each. And applying again that default ratio, conversion ratio, we find that neither meets the 500 threshold. So the indicator value gets an indicator value of 0. However, the genetic evidence reveals a slightly different result. One of the 2 is actually showing a higher effective population size. And so the indicator value in this instance would be 0.5. Both methods are valid, and both have their inherent assumptions within them. And therefore, in this particular instance, averaging the 2 values was deemed to be the most appropriate. So the overall indicator value for the species is 0.25. So that is how this assessments were done. And now what did we find across each of the 9 countries and overall? Well, we were able to assess over 900 species across a wide variety of taxonomic groups, conservation status, habitat types, and life histories. And this amounted to over 5,000 populations being assessed in a relatively short amount of time, just a few months, which is a huge accomplishment, if I do say so myself. Of the populations assessed, the vast majority did not have any estimates based on actual genetic data. And so that's the portion that you can see in the green. That was the portion that actually was acquired from genetic studies. So it shows that genetic data isn't actually needed in order to calculate this indicator. And also just to note that no digital sequence information was accessed or needed in calculating this indicator. The information was simply taken from what was publicly available in literature. Indeed, most of the data used to calculate effective population size was from proxy data, and specifically census size, either as point estimates or broad ranges. And this was a great outcome, as it clearly showed that data was available. So when we looked at the actual indicator values, we found that the majority of species had all of their populations below the critical threshold of 500, or at least the vast majority of populations of species had most of their populations below 500, which was quite worrying. And this was distributed evenly across both plants and animals. However, the opposite was found for the complementary indicator, the proportion of populations maintained. The majority of species tended to have all of their populations maintained, and we see that again, this pattern was consistent across both plants and animal species. And while it's easy to immediately think that our results are likely an artifact of the species we chose, I just want to remind everyone that species across different threat statuses, so across the IUCN Red List categories, were chosen to increase representativeness and an attempt to minimize bias. And so this plot shows that even the unthreatened species, so in particular, the violin plots in green, the dark green and the light green, the near threatened and least concerned, show low indicator values, indicating that they also had populations below the critical 500 threshold. And so they are also experiencing genetic erosion. They have populations that are simply too small to sustain genetic diversity. And in the end, overall indicator values were quantified for each of the 9 countries. And like was shown in the previous theoretical example, these country indicators can be disaggregated, as we see here for South Africa, to get more detailed information on the genetic health of particular taxonomic groups. So to sum up, we were able to assess over 900 species, which included over 5,000 populations in less than a year, and data for at least one indicator was available for over 80% of the species that we looked at. So data is available. It is feasible to report on these indicators. We also found that most populations are too small to maintain genetic diversity, so dedicated restoration and management efforts are needed to enable these populations to increase their NE 500 indicator value. So through these indicators, species and populations that require such efforts can be easily identified before it's too late. And what the complementary indicator showed us is that the majority of populations are maintained, which is very reassuring, but we need to prevent further losses by ensuring that this indicator remains high. And if anyone would like specific details regarding the details of the methods and results, you can access the preprint that is available via the QR code, and hopefully via our published manuscript if it happens within the year. So reporting on these indicators is feasible, and they actually provide a level of sensitivity not currently captured by other species assessments, which is a very novel aspect of these indicators. This information is relevant beyond simply reporting. As already mentioned, it can help identify species and populations in need of management action that might otherwise go unnoticed. They can also help monitor change over time, ideally with respect to tracking the success of monitoring interventions. And these indicators can now help countries set quantitative goals to truly combat genetic erosion. So now we will hear from Dr. Alicia Mastreta Yanez about current and upcoming work to improve the indicator reporting through engagement and with input from parties and others involved in monitoring and reporting.
Hello, can you see my screen and hear me?
We can hear you. We can't see your screen. It's black at the moment.
Can you see it there?
No. Is it just me?
I can't see it in either.
Oh, that's weird. Stop. Can you see it there?
No,
Maybe
do you?
Jess, do you want to share your screen and just advance
Yeah,
the slide?
I was just going to suggest that. Alicia will just tell me when to go next, okay?
Okay, let me use stop sharing. So you are going to share, right? Yeah.
Yes.
Okay, thank you, Jess and Belma. Now I'm gonna let you know that we have been producing a lot of outreach materials, including a policy brief, short videos, and other take-home messages. We invite you to go to the webpage of the Coalition for Conservation Genetics to have a look at the materials that we have produced. Can you go next, please? In particularly, I'm going to stop on the guidance documentation that we made as a follow-up of the co-creation project we did during the first pilot assessment of the genetic diversity indicators that Jess just showed you. So the materials that we have to really train ourselves and to agree on the methods across the 9 countries became this new online documentation that is ready for you to explore, where we have different sections. If you go next, please. Yes. Like a short, quick start list of all the steps that you need to follow in a nutshell. Next, please. And we also have detailed how-to guides with examples, for instance, in how to define populations, how to establish the reference period, how to estimate the effective population size, either from genetic studies or census data, and so on. The idea is that this documentation that is online can be updated as more teams across the world share their experience, their questions, and their ways to assess the indicators. And also because it's an online documentation, it is easier to share a particular step or question, and it's also easy to search and explore using the internet. And this is currently under review, but it has been already published online as a online documentation. Next, please. As part of these guidance materials, we made public a web form that we use to collect data in a more reproducible and standardized way. For this, we use an open software tool called Kobo Toolbox that allows anyone to use the form to create their own project, administer their own teams, change the form, change the questions, and collect data. Think of Google Forms where you have more access and control over the data, and also looks nicer. So we built this form that has different sections for instance, species taxonomy and the data to estimate any 500 indicator. This is also available in the online documentation. As you can see, it says example form, which means that you can click on it, you can fill it with dummy data just to feel how it behaves. And also we made this available for anyone who would like to use it as a template to start their own process, or not, of course. This is all optional. Next, please. Along with these materials, we made available our scripts to summarize data, estimate the indicators, and make some easy plots. This is open access, which means that anyone could use these scripts, and we are looking forward to help people if they get stuck at any moment. Next, please. We also have been working on making the genetic diversity to be included in the National Biodiversity Strategy and Action Plans more easy to incorporate by practitioners. So we have a publication currently in preprint mode, which explains how to set ambitious national level targets, including genetic diversity, specifying capacity and resources needs. Importantly, this preprint connects genetic diversity to other targets beyond target 4 to identify concrete actions, policies, or plans that need to be put in place to conserve genetic diversity. Identifying concrete action, policies, specifying indicators, what data sets, sources, and monitoring programs can be used. Specifically, we also review existing capacities in country, including DNA training programs, laboratories, among others. Next, please. So shortly, our methodologies right now have been improved to make them simple and faster for data entry, clarifying instructions. We have shown case studies, hands-on tutorial, video resources. We are working on translations at several languages. And our idea is that these flows will be integrated into GeoBond processes like bond in a box to calculate the indicators. And also we have other ongoing work to link the data to satellite data, like in the project with Genes from Space, and that is funded by the International Space Science Institute. We're also working with GBIF to reuse the data in a more flexible way. And we also have starting a project called Genamo, which I will explain in a moment, that will also help this co-creation process. Next, please. Genamo is a project that was recently funded. It's a very large, like above 2 million euros project involving the countries highlighted in green in the slide. And the idea of this project, which is led by Dr. Christina Hilson from the Copenhagen Zoo, next please, is to aid the indicator process by determining how to measure the indicator, similar to what we did in the pilot study, but at larger scale and including Earth observation data through an inclusive co-creation process among the countries that I show to improve the workflows. And here I want to highlight that it doesn't mean that they have to use our workflows. Every country is allowed to undertake their own measurements and way to do things. So we hope that Genamo will have countries to include genetic diversity in the biodiversity assessing, committing to the international and national and regional commitments to protect diversity. Next, please. So as a summary of the entire presentation, the genetic diversity indicators do not necessarily need genetic data because census and population data can be used in. They are not that much time consuming to assess. It takes around 1 month and 3 people on average to assess around 100 species. Next, please. We found that there's enough data, at least to assess a little bit more than 100 species per country based on our pilot study. The indicators are flexible, inclusive, and they can save time and money because they use data that is already out there and that took a lot of time to produce, including reports, local knowledge, existing in-country databases, and so on. Next one, please. Importantly, digital sequences information or sensitive information are not needed to be reported. Parties can report, use the indicator value, extracting the data from other studies that can be already published or can be retained by the home institution. The take-home message is that genetic diversity is in a critical situation, and without the genetic diversity indicators, we will not know this. Many populations are too small to maintain genetic diversity, and this means that they won't have enough options to adapt to changing conditions. Therefore, the genetic diversity are useful to raise alarm and to take action. With that, we finish, and we would like to really thank you for your presence during this webinar.
Thank you, Alicia, Jess, and Velma. That's an exceptional amount of information and lots of resources. And as we said in the chat, we will share a lot of the resources with you after the meeting. So look for an email on that with the links to all of the papers and content that you saw. So we're happy to take any questions from the floor. If you wish to, please raise your hand or put it in the chat. You can also ask your questions in Spanish or French if you'd like. So if there's anything that you find you want more information about, please ask away. Francis or Charles, sorry, please go ahead. I'll just allow the mic.
Thank you. Yes, harder to ask without a mic. I put my question in the Q&A, but thought I'd ask it verbally. How do we decide which species to analyze? Because there are only a limited suite of species and highly non-representative for which we have actual population numbers or population counts. And Canada has 80,000 species recognized of wildlife, sort of plants, animals, fungi, and the small subset that we have actual population sizes for, could be completely different from the overall. So an indicator that's a proportion, you'll get a completely different result depending what set of species you choose to analyze. If you only focus on those for which you're concerned, of course, you get a very high proportion with concern. If you focus on a sort of random sample, you could get a completely different result. So I'm wondering how that's being addressed, because I know you focused obviously in your examples on those that we have good data for, and that's not representative of most species in most countries.
So if I can address this, part of the slide, one of the slides that I showed was that bicycle wheel, which showed a small proportion of any based on genetic data, and then most of it based on broad estimates of census. And this wasn't necessarily based on census surveys for the majority of species. It was just even on inferred information. So you would ask someone who has some knowledge of the species if there's likely to be 5,000 individuals, 10,000 individuals. They can give information on a broad range. If less than 1,000, more than 100. So even that semi-quantitative information or estimations are valuable. There was just an amphibian red list workshop that was being held in Cape Town last week, and people who are familiar with certain organisms really do know what the limits of these species can be. So for South Africa, less than a handful of species actually have quantitative genetic or census information. I used one example, but that's one of the only ones. All the other 100+ species, none of this information is known, but we were able to guess and extrapolate based on their experiences in the field, their knowledge of the species in general. And so this can be done even broadly as a start to get an idea of how the populations are doing. I hope that answers it. I'm not sure if Alicia wants to add anything, or Belma.
I just add it only partly answers it, because again,
Okay.
you talked about amphibians, and
Yeah.
that's a tiny proportion, or vertebrates even are a tiny proportion of the animals in most countries. And so, if we limit it to vertebrates or limit it to one particular taxonomic group like birds, yes, we could do that. But how about insects? I mean, what do we know about beetles? What do we know? And in fact, is the ratio of number of adults to an appropriate measure of effective population size for insects even, that 10 to 1. So that's where we can get some very different results depending which taxa we choose. And this is not, yes, this doesn't specify.
Right. So I really encourage you to go through our guidance materials and that Alicia just shared. And yes, there are some areas and taxonomic groups that do need some further attention. So again, this was just a pilot study. We tried to do it, and some groups did look at invertebrates as well. So it is possible, but more difficult. And we are working on that in the ongoing projects to see what we can do and how we can refine things to be more representative across the different groups.
Yeah, just to quickly complement, I think it is important that countries try to have a subset of species that could be representative of the different ecosystem types and taxonomic groups. Of course, this needs to be done. But also umbrella species will be very useful to represent different ecosystems that exist within the country. and some species, of course, don't exist in all of the country, right? So we believe that we have to have a combination of wide-ranging and restricted species from different ecosystems, and also to include species that are important for the countries, because not only wild species need to be incorporated, also domesticated ones to represent our biodiversity.
I'll just address the other hands that I see. So, and then we'll go to the Q&A. So, Jerome, do you want to ask your question?
I've allowed the mic for Jerome.
Now do you hear me?
Yes.
Yes. Jerome from the Federal Office of the Environment in Switzerland. Thank you for the presentation. It was interesting for me. I just had the question. So you had 5,000 population of 900 species over nine countries. So that's quite a few population actually. And do you have recommendation how we could screen much more population? Because to me, yes, 5,000 population. This is tiny. And I'm facing in my daily work. I have to report on the biodiversity overall in Switzerland, and even Switzerland is a small country. But how do you have any ways how we could increase that number of screening many more? Are they technologies or way of doing it? And maybe another, we just had conducted the pilot study in Switzerland on genetic diversity, and I must say that sometimes it's not necessarily true that small populations have few genetic variation. I will be very careful with that statement, just from our pilot experience. But anyway, thank you very much for all these interesting information.
Jess,
So Jess or Alicia?
Yeah, I think we can. I can go first, and then you complement, Jess. Thank you, Jerome, for the question. And yeah, one of the things we are doing we as a whole team of people interested in genetic diversity indicators are to explore ways to scale up this. Especially, we are interested in using Earth observation data and records of occurrence of a species and environmental data to assess population persistence and potentially their size based on habitat size in a more scalable way using praxis. This is a part of a Swiss project we are actually doing called Genes from Space, but also something that Ginamo will be looking at. So I could say keep tuned, because we are really thinking on this. And of course, we have to understand that this will be proxy data, not actual genetic data. But we do believe that this will be better than not having data or ignoring genetic diversity entirely. So a short answer will be yes, this will be scalable using proxy data, and we are in the process of co-developing new methods to do this that could be applied to different parts of the world.
Yeah, and just to add on, I know that some countries, I believe it was Sweden, they have had access to national databases that help them comb through to assess more. I believe it was 20,000 populations, or was that species? I can't recall the detail. So there is ways, if your country has a national species population database, that really helps. Japan also had that for its various plant species. And that was very helpful, and that really helps expedite the assessment of populations. But as Lisa said, there are other avenues that are being explored to do that. Yeah.
Okay, maybe we'll take these next two questions together from Alexander and Belint. So the first is about the example you gave just with the census and effective population size data, do you have any evidence that or estimates yet about how far off the census-based estimates can be for the effective population size? Has anyone done any analysis or level of discrepancy that that's available? And to follow up, perhaps maybe there are some issues with the directionality of the effective population indicator. So for example, an increase in the value does not necessarily mean an improvement for genetic diversity. This can happen in the case of extinctions or fragmentation. Do you plan to do some sort of sensitivity analysis for the indicators?
Yeah, but that's it. Those are great questions. Going back to the example that I provided, there's inheritance assumptions in both types of data that were presented. And so, and depending on the species that you're looking at, the little bit of literature that does exist does indicate that it can differ quite significantly. But there's also been several studies that looked at a range of species that showed that the ratio holds fairly well. Generally between a range of like 0.1 and 0.3 covers a large portion of species. However, more genetic data is also needed. So that will help refine our ratios and comparisons and maybe make them taxon specific ones or species ones. And so if that ratio exists, we highly encourage people to utilize it. And sorry, what was that second question again?
I can answer that.
Yeah, okay, great.
It was like the increase in the value could be an artifact, for instance, if small populations get extinct, then they don't count when you assess the indicator and it gets inflated. This is something we have really thought about it, and the solution, which is explained somewhere in our guidelines, is you have to keep including populations that get extinct in the count of any indicator as 0. So in this way, the indicator value won't get inflated. So an extinct population doesn't become NE, becomes 0 in the count for the NE indicator. And also, we have to be careful with the use of census data for the NE indicator when we have clonal species, of course, because an increase of a number of individuals won't mean an increase in NE if you are cloning them. And this is something that happens a lot in some Mexican examples. And again, this is in our guidelines as short advice. But also I want to say that besides the 9-country pilot study, we are working on individual country, like more detailed explanations of examples there, which will be also published soon. And in these research papers, we will discuss with more detail this type of situations.
Mm-hmm. Alicia, you just touched on a question that was in the chat about how to deal with clonal organisms. So again, is that just something that it's a special consideration?
Yeah, it is a special consideration where you cannot really just count number of adult individuals. You have to count genetes or like group of individuals that are genetically distinct as one. And there are different methods to do this depending on the type of organism, because clonal organisms is a very wide thing. So we are also trying to provide specific advice for this type of situations, but it can be done in some cases, and in others it will be better to assess other type of species. But for instance, in Mexico we have agaves, which is what tequila is made of, and it's a very clonal but also sexually reproducing individual, and there we have to be careful with assessing the number of individuals to make sure that they are not clones.
Just to add also to that, not quite related to clonal species, but for critically endangered or highly inbred species, just an increase in individual numbers and hence census size isn't going to give you a fair look at the effective population size. So the ratios generally apply broadly, generically to species if they are critically endangered, and recovery programs are just breeding inbred individuals. You're simply going to get a reproduction of the same inbred individual, and the effective population size will not increase. So then active management, genetic management of those species and populations will be required to actually see an increase in the effective population size. But these indicators could help flag what species could utilize or benefit from genetic monitoring and focus efforts that way.
Yeah, and that's a good point, because there's a related question. So someone is asking, what conservation strategies would you suggest to take up for improving genetic diversity in a species? So for example, the freshwater crocodile, which shows very low genetic diversity, what can you do to supplement or reinforce or enhance the genetic diversity or support in these types of species? So what type of interventions, I guess, is needed, or is it the natural state of the species and then that's the way it is?
I will say that it has to be done in a case-by-case scenario, and the role of indicators is to point or raise the alarm in some species, and then what to do next needs to be done in a case-by-case. In some species or populations, supplementation can be done, which means bringing individuals from other populations used to increase that diversity and avoiding breeding. But it has to be done carefully and with proper studies. If there are no other species, other populations to reinforce, there are a lot of success stories where very careful breeding, like that means selecting who mates with whom, can literally rescue the species from inbreeding. But this will be extreme situations where there remain very few individuals.
Yes, the cheetah is a great example of that.
Okay, so I think we've addressed we've addressed all the questions either verbally or with written answers. I'm checking in the questions and the chat. Correct me if I'm missing anything, Yvonne. But is there some, yeah. Okay, so yeah, if there's any other questions, maybe a last call. I think we might close the meeting or do something proper. A good way to turn this into a scientific question: how can one ensure that a selection of species will be representative for all species in a country? And maybe the very rich data countries should look at sites for testing. Yeah, like Belen, that maybe gets to maybe the umbrella species.
Yeah, I think this is a very good scientific question that goes beyond the genetic diversity indicators, and indeed more research is needed. But I would like to highlight that we are at a critical opportunity to, for the first time, incorporate genetic diversity, which is really the base of biodiversity, in the monitoring of diversity, which before we were mostly ignoring. So I think we are in a great moment to ask these scientific questions, but to also start taking action, including genetic diversity.
Are there any strategies planned for Asia Pacific regions?
I don't want to speak for those countries, but I know that great efforts have been undertaken with Japan, and I believe there's been some discussions with Indonesia and other countries, but I don't know all the details, so I don't want to speak to them. But many regions from around the world and a lot of different countries have approached to see how they can test these within their countries and stuff. So, and we are working to increase the representation as well. But I believe this is the first step, and it is spreading.
And yeah, there are projects that are being reviewed, so still not funded, but maybe so, where some other Asian countries are included in this international endeavor. And I would like to highlight that I completely forgot about it because I'm forgettable. But there are already guidelines for selecting a species for monitoring genetic diversity published by the IUCN and some of the people that collaborated in this report. So we are not starting from zero in that regard.
Right, and it's just in the chat. Thank you, Kristen, for putting that there.
Yes, and I also put it because I can't access chat for some reason. It says it's not available, but I posted answers and links for the documents in Q&A session, so you can also check it there. And just to supplement, there are also projects already ongoing on behalf of WCMC, which are also encompassing Asian countries to test these indicators.
Great. Well, thank you on behalf of Giovanni. Jillian, are there any closing remarks you want to make?
No, I think this has been really, really interesting. And as I mentioned in the chat, we will be sharing the video online. So feel free to share it with others who you think might be interested.
Okay, thank you. Thank you, everyone, to your questions and for your interest. See you later.
Thank you for the participation. Thank you. Have a nice day. Bye.
Bye.
Bye. Thank you.